step 1, step 1A, step 2, and now we're at step 3. Now we left off last time, not right at the very end of last time, but during our time last time, we talked about all of the electron carriers that we generate in steps 1 through 2, right? In step 1, we generate 2 NADH, yes? In step 1A, we generate 2 more NADH, yes? In step 2, we generate 6 more NADH and 2 FADH2. So we have a total of 12 electron carriers to use in step 3. And so step 3 is, where do those electrons go? And so the question we're going to answer as we talk about step 3 is this one. How are electrons transferred during oxidative phosphorylation? We've already talked about another type of phosphorylation, and what was it? What was our other type of phosphorylation? Substrate level phosphorylation. That's where you take a phosphate off of the substrate, off of something the enzyme is interacting with, and put it on something else. In oxidative phosphorylation, we're taking free inorganic phosphate and sticking it to ADP. We're not stripping it off of the substrate of an enzyme. We're taking free inorganic phosphate and then smashing it onto ADP. All right. So, big picture. Big picture of what's going on in oxidative phosphorylation. Electrons are removed from our electron carriers, and the electrons on those electron carriers are at a really high energy level. Not as high as they were when they were in glucose, which is why they were able to transfer to NAD plus in the first place, or to FAD in the first place, but they're at a really high energy level. And they pass through a series of intermediates every time dropping energy level, because that's what electrons want to do, right? Whether you're in Gen Chem now or not, hopefully by this point you've taken some chemistry class and you know electrons want to be in the lowest energy orbital possible. That's what electrons want, and that's what drives most chemical reactions. And the way I say most is because we can force electrons to do whatever we want, but most chemical reactions are fueled by electrons dropping to a lower energy level, because that's what electrons want. All right? Now, this process of electrons transferring from our electron carriers ultimately to oxygen is a spontaneous process. It's a spontaneous process, and it releases free energy. Free energy that the cell can use to fuel processes that require energy. What are some processes that we know cells do that require energy? Give me some of these. Okay, so you're talking about you're talking about putting a phosphate group onto a pump. Yeah. But yeah, what do pumps do? They use ATP to do what? To actively transport things against their concentration gradient, right? That's something a cell does that requires energy. And many of those pumps, like the sodium potassium pump, is also an ATP hydrolyzing pump. So it hydrolyzes ATP as it fuels active transport. But you can use anything to fuel active transport. It doesn't have to be ATP, right? You just need energy in order to fuel it, because it's not fueled by diffusion. It's not fueled by a concentration gradient. And so as these electrons drop and they release free energy, that energy is used to actively transport hydrogen ions into the inner membrane space. So in your mind right now, you should have an image of a mitochondrion. Do you have an image of a mitochondrion? How many membranes are there? 2. Explain to me the inner mitochondrial membrane. Yeah, it's all folded and wrinkly, right? Tons of surface area, folded, a lot of surface area for stuff. And what's in between those two membranes? Space. And you know it's inside of a cell, and cells are water-based, so you know that that space inside there is water. Water is the solvent, but there's space in between those two membranes. And space is something that you can use, right? The mind of a hoarder. Space is only valuable when it's full of stuff, right? And so we've got this intermembrane space, and it's only valuable if the cell packs hydrogen ions into it, because then that sets up a really steep concentration gradient, right? And now that space is valuable. And so that's what goes on. And then so as we look through this, the electrons from NADH enter into this complex, or enter into the electron transport system at complex 1. Whereas the electrons from FADH2 enter into complex 2 and then pass from 3 to 4. And I'll show you what that means as far as the amount of energy generated. The amount of energy generated in this process. Yeah.
So how the mitochondrion works with
Yes.
two membranes and all that, how does this explain, let's say mitochondria was a free living organism at one point that was
Alpha proteobacteria is the idea.
Is that how alpha proteobacteria even work?
No, but they do have it's not, because the idea is that the outer mitochondrial membrane came from the cell that phagocytized the alpha proteobacterium, right? The alpha proteobacterium didn't have two membranes. That second membrane came from the cell that phagocytized it.
Yeah.
And so you can only work if you have the two membranes and you've got that intermembrane space.
So did they even have that complex 4 protein originally?
That's a good question. I don't know. I don't know the answer to that question. My guess would be that they do not. However, I can tell you that the membrane of alpha proteobacteria is very similar to the inner mitochondrial membrane. It's structurally similar. It's got a similar ratio of phospholipid types, and it's kind of wrinkly. Yeah, so there are some similarities there. All right, there's some added complexity, right? You're like, this is way too simple. I want more. I'm hungry. Give me some more. Here it is. So it's not just, electrons aren't just going from the electron carrier to complex 1, to complex 2, to complex 3, to complex 4, and then to oxygen. They're actually being shuttled in between complexes by moving down energy levels as well. And so you have molecules, cytochrome C and ubiquinone, also known as coenzyme Q, that actually are in between. They're in the inner mitochondrial membrane, physically in between the complexes, to allow the electrons to continue to drop down energy levels and to move from complex 1 to complex 2, and complex 2 to complex 3, and complex 3 to complex 4, and then complex 4 to oxygen as the final electron acceptor. All right?
Why is it called a heme group if it's iron? Like, why is it called an iron group?
Why isn't it called an iron group?
Or a Phe group?
I don't know. I don't know the origins of the name heme. I don't know what it means. I mean, you find it in hemoglobin, it's an iron-containing group, and you find it in myoglobin, and it's an iron-containing group. I don't know the meaning of the heme, I'm sorry. All right. So specifically because hydrogen ions, you're like, well, this is cool. We're actively transporting something, right? You're like, okay, I get that. And it needs to be fueled by energy. And a lot of pumps use ATP. But you're like, okay, if we're releasing energy from these electrons, I can see how we would use that energy to actively transport hydrogen ions, right? Can you see that? Maybe? Even if you can't see it, can you imagine it? Can you feel it? Most importantly, can you taste it? Right? Hydrogen ions taste sour, by the way. Anyways, that's another story for another time. But the hydrogen ions, you've got a problem. Ions cannot move through a lipid bilayer. So you're like, cool, we're using energy to actively transport hydrogen ions, but we still need a channel. And so what you find is that complexes 1, 3, and 4 are ion channels. And so we've got the energy to actively transport it, right? And where's that energy coming from? Where's the energy coming from to actively transport hydrogen ions? It's coming from the electrons as they drop to lower orbitals. Okay, it's coming from that free energy released. Okay, we're like, okay, we've got the energy. And now we've got the channels that we can move hydrogen ions through, because you cannot move ions through a lipid bilayer. Notice what's missing from this. Which complex is not a hydrogen ion channel? Complex 2. So if FADH2 gives its electrons to complex 2, instead of complex 1, true or false? FADH provides less opportunity to pump hydrogen ions against their concentration gradient. True. Less opportunity, right? And what you're going to find is less hydrogen ions means less ATP generated. Okay? So less hydrogen ions pumped against their concentration gradient is going to equal less ATP produced. So true or false? FADH2 is going to produce less, right here, I was pointing to it right in the first place, less ATP than the electrons on NADH. True or false? True. Okay? It's an important thing to keep in mind. Yeah, Charles?
What's the benefit of FADH then?
That's an excellent question. So if you look where FADH is generated in the Krebs cycle, it's generated by an enzyme that, one, cannot bind NAD+. So that's one issue. But it's also taking electrons that are starting at a lower energy level than the electrons that are transferred to NAD+. So it's still making the most of what it has. It just doesn't have as much. Right? That would be like you asking my parents, what was the point of having a second son when you already had Dr. Engel? Which they didn't call me Dr. Engel when I was a kid. You better believe they call me Dr. Engel now. No, I'm just kidding. That would be so terrible. Mom, you cannot call me Matt. You need to call me Dr. Engel. I'm sorry. This is just ridiculous. They don't call me Dr. Engel. My kids do. But my parents don't.
When they're trying to get your attention?
No, I don't think my kids know I'm a doctor because it would confuse them. They'd be like, well, then why do we go to the doctor for a checkup? If Dad's a doctor, I don't want that confusion. So, well, I don't know even what happened there. We got really onto a really crazy topic. That was my fault. I apologize.
So
yeah, Rachel.
The last point. What's the last part? Like, there's something weird about the wording.
Oh, so the hydrogen ions are shuttled through. So the ion channels complexes 1, 2, and 4. The one above it. Cytochrome C and ubiquinone shuttle electrons between the complexes when Heem's groups on the enzymes accept and donate electrons.
Okay.
Or you can just eliminate the ion. I'm sorry. Yeah, that's terrible too. That was probably worse than my little rant that just happened. All right? Let's have a lecture break. Y'all ready for a lecture break? I'm not quite ready for one, but we're going to do it anyways. Okay? So what I want you to do is this. As you work with the members of your group, use the information that we just discussed, that FADH2 is going to produce fewer ATP than NADH is going to. Use that information to try to approximate the amount of ATP that we're going to produce in this electron transport chain. Keeping in mind that we can't produce a fraction of an ATP molecule, right? You either produce an ATP molecule or you don't. You don't produce half of an ATP molecule when the whole process is over, okay? You may produce half per, electron, but you're not going to produce half overall. So don't use anything else. Don't use your notes. Just use your logic and the collective intelligence of your group to try to figure out the number of ATP that are going to be generated from 10 molecules of NADH and 2 molecules of FADH2, because that's what's bringing electrons to the party, right? Okay? Work through this a little bit, see what you come up with. Don't use anything except for what you can think of and what the people around you can think of. 2 minutes, starting now.
Right. Here's another thing. 12 carriers, each carry 2 electrons. 24 electrons. Which I think would make sense that every electron would transfer one hydrogen ion. The one hydrogen ion per electron. So we transfer 24 hydrogen ions. And this would make sense that when those
Oh, gosh.
for every diffusion channels, they call it that makes the ATP. So every time that one hydrogen ion would fuse, so I think you get 24 ATP.
Yes. So try to come up with an approximate number of ATP generated from 10 molecules of NADH and 2 molecules of FADH2. Keeping in mind that overall in the process, we cannot generate fractions of ATP. Okay?
So the only problem is instead of
And how many FADH?
then again, every time that electron changes, energy
And also remember the electron has been transported and changes orbital position. That's what generates energy that pumps ions across its gradient. And then it comes back to the last complex because the gradient there's less hydrogen ions inside the mitochondria than outside the inner mitochondria. That's what energy comes on to combine phosphate. ATP.
Yeah.
So it's actually not the electrons themselves that generate phosphorus, but it's actually ions going through.
Oh, well, I'm not the only person that's done this. Look at all
but the
this.
energy's still coming from
Mark's too fast. I ran out of space.
So it's basically it depends.
No,
Oh,
that's a wall.
Yeah, but somebody else has done that too because it's dirty.
Not to write on the wall, Doctor.
You could just made the line smaller.
That's an excellent question. I don't know. I don't know. I don't know the answer to that question. All right. So here we are. Here we are, okay? So we're trying to figure out what we're going to do in step 3. We've already established that NADH is going to produce more ATP than FADH2 is, right? We already established that? We good? And we're at this point as well because we know what we've generated in steps 1 through 2. In step 1, we generated 2 net. Are you guys laughing about writing on the wall? It's fine. Relax. I'll clean the wall. Actually, I won't clean the wall. 2 ATP, but we also produced 2 electron carriers. In step 1A, which is pyruvate oxidation, we didn't produce any ATP, but we produced 2 more electron carriers, right? And then in step 2, we generated 2 more ATP, 6 more NADH, and 2 more FADH2. So 8 more electron carriers. And now the question is, what are we going to do with these in step 3? So here are your options. Here's what's nice. We're dealing with even numbers, right? 6 and 2 and 2 is 10, and then 2 is 12. Which means even if each only generates a half of an ATP per molecule, we're still going to overall get to a whole ATP, right? Because we're dealing with even numbers. And a half times any even number is going to be a whole. If you don't believe me, try it out. Sit there and do 1/2 times 4, 1/2 times 6, 1/2 times 488. 1/2 times 1 is not it's not an even number though. 1 is an odd number. So, okay. So any even number, we're going to get a whole. So then you're like, well, what do we do here? Let's assume that each NADH is worth how many? How many do we want an NADH to be worth?
2.5.
Why are you going to use 2.5, Chad? Where'd you get that number?
That's just a guess.
Yeah, right.
Great guess.
That was a great guess, because that's actually what NADH is worth. Each NADH is worth 2.5 ATP. Okay, each NADH is worth 2.5 ATP. How many NADH are bringing electrons to the party in step 3? 6 plus 2 plus 2. 10. Okay, what's 10 times 2.5? 25 ATP. Okay, from the NADH. Okay? We also have 2 FADH2. We already said they generate less ATP than NADH does. How many do you want these to be worth, Chad? 1.5. So what's 1.5 times 2? 3 more. What's 25 plus 3?
28.
28. Nicely done. Joey, 25 plus 3. You can't even find that in any kind of a table. That's got to be here or here. Okay? So we generate 28 ATP in this process. We also generate 10 molecules of NAD+, and we generate 2 molecules of FAD. And that is a very important point to illustrate, because what are these? These are things that need to be reduced to become these, right? And now they're regenerated, and it's not recycled, they're reused. Because if they were recycled, they would need to become something else, but they're reused so that you can continue this process. Isn't this wonderful? Okay, so in step 3, we generate 28 ATP, so plus the 2 here, plus the 2 here, and what do we get? 32 total. 32 total ATP for every 1 molecule of glucose in most cells. And some cells don't have FAD, and they force the electrons to actually go up to NADH. Even though they wouldn't normally do that, they force it. And so instead of having 10 NADH and 2 FADH2, you actually have 12 NADH. Is that right? Yeah. You have 12 NADH. And what's 12 times 2.5? 30. So instead of generating 28 here, we would actually generate 30, which would make this total up to 34 instead of 32. But that's just bonus information. That's just bonus. You're not going to be held responsible for that. All right? Yeah, Joey.
So why is it that when we read the book, it said that the amount of ATP produced varies significantly, and it didn't give us a number like this?
Because there are cells that don't have FAD. And so it'll vary. Instead of 32, it'd be 34. And then there are some that actually are a little bit more energetically efficient, and they trap additional electrons. And so there are some cells that can generate as much as 38. But it's not as neat, and it's not as structured. So I'm giving you the neat and structured and saying, assume this is always the case, but no, it's not. Right? Not N-O, it's not, but K-N-O-W, it's not. So assume this is always the case, but know in your heart that that's not true. That there actually is quite a bit of variation. Yeah, welcome to biology. Yeah.
So is there a difference between like glucose and glycogen being broken down with the ATP?
No, not really. So breaking down glycogen into glucose is a spontaneous process and doesn't require energy investment. It does require enzymes, but it doesn't require energy investment. And so no, you'd still get it down into individual molecules of glucose that could run through it.
Because one of the slides from another class was saying it's 33 to 32.
If
Differing with like glycogen and glucose.
with glycogen and glucose,
Yeah.
that was from our text,
No, from other things.
from like a human physiology textbook.
Yeah.
Yeah, well, so if it's 33, what that would mean is that when you break down glycogen, just in breaking those bonds, you're able to trap some of that energy and use it to synthesize ATP. Yeah, but it's not gonna change this process here. All right. So this part we already talked about, so I kind of just put the next slide in there and put a lot of information, because this we've already talked about. In complex 1, 3, and 4, and in ubiquinone, we pumped hydrogen ions against their concentration gradient, right? And so we set up a really steep concentration gradient. We also set up a voltage gradient. Because if you pump a lot of material into the inner membrane space that are positively charged, what does that mean about the environment in the inner membrane space? How is it charged? It's positively charged. What does that mean about the matrix? If you pulled a bunch of positive charge out of the matrix and force it into the inner membrane space, what does it mean about the matrix? Negatively charged. So not only do hydrogen ions want to move into the matrix because they would move down their concentration gradient, they also want to move into the matrix because there's negative charge there and they're positively charged, and positive charges are attracted to negative charges, right? Opposites attract. And so the hydrogen ions really, really want to get into the matrix. But they can only do so by going through a channel. And the only channel that allows them to do that is an enzyme called ATP synthase. That's the only thing that allows hydrogen ions to move from the inner membrane space into the matrix. And as they do, it synthesizes ATP. Yes, sir. At a rate of 2.5 per NADH and 1.5 per FADH2. Yeah.
So that's the rate of like NADH per speed. Do we know the rate of like electron per?
Of what it is per electron?
Yeah.
Well, each NADH is carrying 2 electrons. So if it's 2.5 per NADH, it's 1.25 per electron.
Okay.
Right? For the electrons here. But then the electrons here are only worth 3/4 of an ATP. So that shows you that the electrons are starting at a lower energy level.
Okay.
And so that's why they can't drop as much and release as much energy. All right?
Are you gonna do?
I'm going to move to the next slide. Oh, yeah. It's time. And so here is this enormously steep hydrogen ion concentration as they are shuttled across complex 1, complex 3, complex 4. And notice a little bit about the arrows aren't illustrating energy level of the electrons. They're just relative position. Okay, so it's a little bit confusing. So relative position, not energy level. This drop here from this cytochrome inside of complex 4 down to oxygen is a huge drop. A huge drop in energy. And what do you know about huge drops in energy? They release a lot of energy. And so going back to this previous slide here, came back, if you want it again. This energy is used to actively transport materials into and out of the mitochondria. What material, the only material so far that we've mentioned being actively transported into the mitochondria, what was it? I'll give you a hint. It's the product of step 1. What's the product? Pyruvate. Pyruvate is the only thing so far that we've mentioned being actively transported into the mitochondria. The only way that that is possible is if you have oxygen present to act as the final electron acceptor. You get this huge drop in energy as electrons move from complex 4 to oxygen. That's the only way you generate enough energy to actively transport pyruvate into the mitochondria. Which means you only actively transport pyruvate into the mitochondrion when what is present? Oxygen. So if oxygen isn't present, pyruvate does not get into the mitochondrion, which means step 1A doesn't happen. Which means you can't go through cellular respiration. You go through glycolysis followed by fermentation. All right? So what does that mean about where fermentation happens? It happens in the cytosol because pyruvate did not get into the mitochondrion, so fermentation has to happen in the cytosol in the same place pyruvate is being generated. And this is true whether you have ethanol fermentation or lactic acid fermentation or lactate fermentation. So if you are a yeast and you have ethanol fermentation, where's the ethanol being produced? In the cytosol. Yeah.
So, there's some bacteria that they don't even use oxygen at all.
Oh, sure. Yeah, there are organisms that aren't just bacteria. A lot of parasites, especially a lot of intestinal parasites, don't use oxygen at all.
Even though they have a mitochondria, or?
Yeah, they have mitochondria, but a lot of times they don't transcribe the mRNA that would be translated into the machinery of oxidative phosphorylation. So even if oxygen was present, they wouldn't use it. So they have mitochondria, but they don't use them in that way. Yeah. But that's because they came probably from a free living ancestor that needed oxygen. All right? Any other questions? So I'm going to skip the last set of slides that talks about fermentation when oxygen isn't present, because I just mentioned to you, and so it's on the recording, you can access it again on YouTube, that the pyruvate, which actually ends up, fermentation is pyruvate reduction. You actually force electrons onto pyruvate from NADH to regenerate NAD. No, let's go through it. It's worth it. It's worth the time. It's worth your time, it's worth my time. What about when oxygen is absent? I know what you're thinking there. You're like, this is not worth my time. But you're here. So your actions say differently than your thoughts. Right? All right. Did you say okay, Mom? All right. So when oxygen isn't present, and we call these facultative anaerobes, so those are organisms that can operate in the absence of oxygen, but typically don't. Some of our cells are facultative anaerobes, some are not. Okay, our muscle cells are facultative anaerobes. They can operate in the absence of oxygen. So the electrons passed to NAD+ during glycolysis have to be transferred back to a different electron acceptor. So glycolysis, when oxygen is absent, continues to generate ATP through substrate level phosphorylation, right? As phosphate groups are ripped off of the substrate of the enzyme and attached onto ATP, or ADP to form ATP. So glycolysis still continues. Step 1 generates 2 ATP. That continues. And then pyruvate actually acts as an electron acceptor. So it gets reduced and gets converted into either lactate or into ethanol. In our cells, it gets converted into lactate. In a yeast cell, it gets converted into ethanol. So glycolysis continues, and it generates 2 pyruvate for every 1 glucose. And each of those pyruvates can act as an electron acceptor for NADH to give its electrons back. I guess it didn't take them from pyruvate, but it took them from the molecule that generated pyruvate. It gives them back to form either lactate or ethanol. And so here's lactate fermentation. So glycolysis happens, generates ATP, and then NADH gives its electrons to pyruvate to form lactate, or lactic acid. Lactate is the conjugate base of lactic acid. And so this is the fermentation that happens in our cells. This is the fermentation that happens in yeast and other eukaryotic cells that are ethanol fermenters. All right? Any questions, thoughts, concerns, things you feel like the class as a whole could benefit from hearing before we transition over to chapter 8? Yeah, Rachel.
Back a couple slides because I missed the last part of the one equation that was up there. That one.
Do we need to know that equation for anything?
Do we need to know that equation for anything?
I mean, like a test or...
I don't know yet. Yeah, I don't know yet. I would say, at this point, I would say yes. Would maybe end up being no? Sure. But that's a better situation than for me to say no and it end up being yes. Because you'll be disappointed if I say yes and it ends up being no, but you would want to kill me if I say no and it ends up being yes. I have disappointed more people than I can ever list to you in the amount of time we have together. As far as I know, there's nobody that wants to kill me, other than my brother. But we're okay now. Because I know how to tell when he's coming. I'm just kidding. He doesn't want to kill me. I don't think. All right? Man, this is fun. I've been having fun today. I hope you've been having fun. I know. I was really mean to my brother. I guess you could do that now, but don't. All right. So as we start talking about chapter 8, we need to split photosynthesis up in the same way we did cellular respiration. In cellular respiration, we split it into steps that we can use and relate to and deal with one at a time. We have to do something similar in photosynthesis. But before we do that, we have to talk about big picture stuff. So what happens overall during photosynthesis? How is water used during photosynthesis? Where does it happen? And then how does the entire plant function in photosynthesis? So what are the photosynthetic parts of the plant, and how do the non-photosynthetic parts aid the photosynthetic parts in carrying out photosynthesis? Glad you asked. That's a good question. We'll deal with that. So what happens during photosynthesis? Big picture. Here's the reaction, the overall reaction of photosynthesis. 6 molecules of carbon dioxide and 12 molecules of water used to generate one 6-carbon sugar, but actually photosynthesis only develops 2 3-carbon molecules. But then anyways, and then 6 molecules of oxygen and 6 molecules of water. Yeah.
Why don't we write 2 of the 6?
I don't know. I think it's because we want photosynthesis to be more clearly the exact opposite of cellular respiration.
But it's not.
It's not.
Why don't we just write it?
I don't know. Do you want me to write it that way? I'll write it that way from now on. I will never again use this equation, but I don't want to change it right now. So overall, carbon gets reduced during photosynthesis. During cellular respiration, carbon got oxidized, as fully oxidized as carbon can get, since carbon does not ionize. And in photosynthesis, carbon gets reduced to as much as carbon can get reduced. Again, it does not ionize, and so we can't force carbon to take on a positive charge, which you would really want to if it was going to get fully reduced, right? You would want it to take on a negative charge. Did I say positive charge? You would want it to take on a negative charge if it got fully reduced, but it doesn't. So we're going to talk about as reduced as carbon can get is when it's sharing electrons with something else. You're like, well, what about if carbon were just by itself with its electrons? But if carbon were just by itself, it wouldn't have 8 electrons in its valence shell. It would only have 6, and it's unhappy. It's unhappy. Unless, of course, it crystallizes, which it will to form diamond and graphite, but only under high pressures. Really weird situations. All right. So what we're doing here is carbon dioxide ultimately gets reduced into some kind of a carbohydrate. And carbohydrates always have the empirical formula of CH2O, and then you can have a certain number of those CH2O components to make up as big of a sugar as you want to make. I mean, you could literally have billions of these pieces all strung together. And many cells in your body have that right now until they work through their glycogen stores in between meals.
Is the CH2O ratio just due to the fact that it's reacting with water? Is that why the H2O is going to be in that same ratio with O?
Yeah, I mean, sort of. Yeah. The oxygen here, though, didn't come from water. It actually came from carbon dioxide. So it's a little confusing. All right. So again, we can split photosynthesis into 2 parts: the light dependent reactions. And what does it mean to be light dependent? You need light. And then you have the light independent reactions. What does it mean to be light independent? You don't need light. Does that mean it does not happen in the light? No, it just means that it does not require light to happen. There are some plants in which it doesn't happen in the light, but for most plants it happens all the time. Yeah.
But isn't that kind of not true because it needs the light dependent to be able to be light independent?
Well, I mean, I suppose you could look at it that way. But that would be like you arguing that I don't need plants in my diet because I eat animals that eat plants. But I really don't need plants in my diet. I take a multivitamin, I get what I get, and then I just eat meat. And it's awesome. I eat meat and sugar.
So it's like saying a plant-independent person, they actually do need plants.
Right, but it's still a legitimate feeding strategy. I'm still a carnivore, right? Because I don't eat plants. I'm not an omnivore. That's not true. I do eat plants, but it was funny in my head. It didn't. I guess maybe it wasn't delivered well, or maybe it just was legitimately not funny. I don't know. Oh, gosh. All right, so here's just a synthesis of what's going on during the process of photosynthesis. So in the light-dependent reactions, we take ADP, an inorganic phosphate, and we turn it into ATP, and we take an electron carrier, and we reduce it by putting electrons onto it and a hydrogen ion, just like we did with NAD+, except for this is chemically a little bit different. A little bit different. Oh gosh, speaking of the funny but not delivered well, I don't know if that was the case or not, but my brother-in-law, oh gosh, I shouldn't say this because it's being recorded. Anyways, that's fine. My brother-in-law yesterday preached, and he had some amazing jokes because I read his manuscript ahead of time, and they were amazing. I laughed as I was reading them. But then he was a little bit nervous, so he didn't deliver them well, and I felt so bad because they were so funny. But then he didn't deliver them well, so only I laughed. And he laughed too, but, it's not the same. I feel like I have the opposite problem, where, like, my jokes legitimately aren't very funny, but, like, I'm awkward and, like, chubby, so it's like, oh, he's just so funny, It's like, no, he's really not. It makes you a little bit uncomfortable, so you don't know what to do other than to laugh. Right. All right. Thank you, Joey. That was awesome. All right, so how is water used during photosynthesis? How is water used during photosynthesis? So ultimately, this is what happened. If we're going to produce carbon, we've got to oxidize something else, right? If you're going to reduce something, you've got to oxidize something. Ultimately, water is oxidized in order to allow us to reduce carbon. If we're going to add electrons to carbon dioxide, we've got to take those electrons from something else, and we take them from water. We take them from water. And you're like, well, where does sunlight come in then? Isn't sunlight providing the energy? Why is it the electrons? What's going on here? Glad you asked. We'll deal with that when we deal with the light-dependent reactions of photosynthesis. So here's what's going on. We take 2 molecules of water, we strip it down into a molecule of diatomic oxygen, molecular oxygen, 4 hydrogen ions, and 4 electrons. So every water molecule that you split, you get 2 hydrogen ions and 2 electrons. But you can't just split one water molecule, because then what do you do with that free oxygen? That free oxygen is very, very dangerous. I don't know if you know that or not. Have you ever wondered why people say it's healthy to have antioxidants in your diet? It's because those free oxygen radicals are really, really dangerous. Okay? But if you split them 2 at a time, then the oxygen, they just pair together, and it's molecular oxygen. It's not dangerous. Unless, of course, there's a spark, then it is really dangerous, because then it feeds fires. And, oxygen is pretty much always dangerous, but we need it. So water provides the hydrogen ions for ATP synthesis, right? We saw in cellular respiration, we took hydrogen ions and we pumped them against their concentration gradient. Or if you're going to make ATP during the light-dependent reactions of photosynthesis, what do you imagine happens? The same thing. We take hydrogen ions, we pump them against their concentration gradient, and we allow them to flow down. And when they do, they take inorganic phosphate and they attach it to oxygen. And you should have predicted that when you saw in the previous slide that we took NADP. We took ADP. We took ADP and an inorganic phosphate. So you're like, this isn't substrate level phosphorylation, right? Because substrate level phosphorylation, we rip a phosphate off of something and put it onto ADP. But this was an inorganic phosphate. So as soon as you saw that, you should have been thinking, that looks just like what happens during oxidative phosphorylation, step 3. And it is. But we're out of time. So we'll pick up here on Wednesday. Have a wonderful conclusion to your Monday. If you have lab this afternoon, have fun feeding yeast and watching them produce booze and carbon dioxide.